The gastrointestinal tract is the site of where ingested foods and drinks are absorbed into the body. This provides opportunities for several external microorganisms to enter and invade the GIT, some of which are pathogenic. These pathogenic organisms of course induce a GIT illness.
Erymonas hydrophilia is a facultative anaerobe that can be found in fresh produced meats such as beef, poultry or pork, and dairy products including raw milk. Different agar plates are used in the initial analysis of the organism. On the horseblood agar, there are large cream-coloured colonies that are surrounded by a double zone of hemolysis, which is a clear indicator of this organism. The McConkey plate displays large, pink, irregular-sized colonies indicating a lactose fermenter. The CIN agar plate is used to determine Erymonas or Yersinia species. In this instance, a clear Erymonas species is identified by the red bullseye center of the colonies surrounded by a translucent zone. A positive catalase and a negative oxidase test plus an API 20Ne are used to confirm the presence of Erymonas hydrophilia. Vibrio parahemolyticus is another facultative anaerobe that is the leading cause of GIT infections. This is associated with the consumption of raw contaminated shellfish and oysters. Horseblood agar is used in the initial analysis and displays large creamy colonies which emit beta hemolysis. There are small yellow colonies on the McConkey agar indicating a non-lactose fermenter. A TCBS plate is used to differentiate Vibrio species, with this organism having large colonies with a blue-green center. An O129 disc that is sensitive also indicates a Vibrio species. A positive catalase and oxidase test, as well as an API 20Ne, are used to confirm the presence of Vibrio parahemolyticus.
Salmonella food poisoning is usually associated with food containing eggs, meat, and poultry. It is part of the normal flora in the gastrointestinal tract of many animals, commonly associated with chickens. Contamination occurs when these foods have not been adequately prepared or stored and are then ingested. There are three main agar plates used to distinguish Salmonella: McConkey, horseblood agar, and XLD. Salmonella chrome agar can also be used as an identifier. On MAC, the colonies are yellow as they do not ferment lactose. They are small and white with gamma hemolysis on horseblood agar and grow in pink colonies with a black center due to H2S production on the XLD agar. On the chrome agar, these colonies will appear purple in color. The other test results include catalase positive, oxidase negative, and gram staining indicates a gram-negative rod. An API 20E can also identify this bacteria. Shigella contamination occurs via the fecal-oral route and is transmitted person to person. It is usually due to poor hygiene practices of food handlers. The plates used to distinguish Shigella are McConkey, HbA, and XLD. On McConkey, these colonies are yellow as it does not ferment lactose. The cultures on HbA are small, white, and have gamma hemolysis, and on XLD the colonies are pink. Other distinguishing tests are catalase positive, oxidase negative, and are gram-negative rods when stained. An API 20E can also be used to identify Shigella.
Yersinia enterocollitica is an organism that is usually found in the feces of both wild and domesticated animals. Infection therefore occurs from the ingestion of improperly cleaned food sources. The main agar mediums used to identify this organism include HbA, McConkey, and CIN. Gamma hemolysis is apparent on HbA. Yellow colonies are seen in McConkey indicating non-lactose fermentation. Tests that are vital for identification were the red bullseye-like colonies on CIN agar that are a unique characteristic to Yersinia species, as well as a gram stain morphology of gram-negative rods that are catalase positive and oxidase negative in addition to an API 20E. Edwardsia tarda is an organism that is usually found in the GIT of fish in humans. Infection is usually the result of ingestion of contaminated food products. The biochemical properties closely resemble Salmonella species in that it is a gram-negative rod and oxidase negative. Gamma hemolysis is seen in HbA, pink lactose fermenting colonies on McConkey, and pink colonies with black centers on XLD, suggestive of H2S production. Edwardsia can be distinguished from Salmonella by the lack of mauve colonies on chrome agar, as well as an API 20E and having a negative catalase test.
Case 5 showed signs of invasive gastroenteritis, with the 25-year-old male having fever, cramps, and diarrhea. A loose stool sample had moderate levels of RBCs and pus cells. The pure culture of the specimen contained GNR bacteria with distinct S shape. Already we were suspecting Campylobacter species due to its unique morphology. Moreover, the organism was catalase and oxidase positive and presented signs of alpha hemolysis, albeit its small zone. The TCBS plate grew no colonies from the APW. This was also evident for the XLD, MAC, and chrome agar for Salmonella plates cultured from the MSB. As a result, Salmonella, Shigella, and Vibrio species can be eliminated as possible causes. Fortunately, the organism grew in the CCDA plate incubated at 42 degrees Celsius, which is highly selective for the suspected Campylobacter. All that was left to do was to identify its species. This was achieved through a Hipurate test and a cephalothin disc which had positive and resistant results respectively. Campylobacter jejuni was identified as a pathogenic agent. On the other hand, case 6 is a non-inflammatory infection based on clinical notes of a 35-year-old female employed at a reptile farm. This also helped us begin to suspect causative agent, namely Plesiomonas. Moreover, the pure culture produced catalase and oxidase positive GNR bacteria with no hemolysis. Again, there were no colonies growing on the plates inoculated by the APW and MSB, indicating an organism other than Shigella, Salmonella, and Vibrio is responsible. However, it is important to note that the colonies incubated aerobically and at CO2 were similar, thus indicating that the organism was a facultative anaerobe, a distinct marker. The conspicuous colony morphology was also unique. The API-20E confirmed the identity of the organism as Plesiomonas shigelloides.
An example of a doctor's report after GIT pathogen identification.